this is the steps of mitosis, and we will be keeping track of the numbers of chromatids and chromosomes and centromeres in one pair of chromosomes as we go through the steps. This is for Dr. Olga Kopp's class, and this is for students Justin Ewell and Stephen Green. First, we have prophase, and in prophase we will have the centrioles and centrosomes will start to form the early mitotic spindle with the microtubules, and we will also have the nuclear envelope, and inside the nuclear envelope we will have two chromosomes, or one pair of chromosomes, and each chromosome is made up of two identical sister chromatids, and each chromatid has a centromere, as you can see there. Not the best drawing, but you get the gist. This is kind of the breakdown right here. The two identical sister chromatids are joined together at their centromeres and all along their arms by cohesin. So those together equal one chromosome. And then the two individuals there equal, there's two chromosomes total, making one pair. And we have that right down here. Total inside there is one pair of chromosomes, two pairs of sister chromatids, and four centromeres. Otherwise, there's, you could also say it as two chromosomes, four sister chromatids, and four centromeres. Okay, and from there, the light will obviously be blocking this, so we can't see it. So we're going to go ahead and try and move out of the light here, if I don't make you too sick. Okay, next is prometaphase. It's kind of like an in-between step. You're going to have fragments of your nuclear envelope that will be kind of spread throughout. And you're going to have, again, your centrioles and centromeres with the microtubules that will be coming off them. They will be now looking to attach to the centromeres on those chromosomes. The centromeres of each of the individual sister chromatids. And on those centromeres, there will be kinetochores, and there they are right there, or kinetochores, however you want to say it. And there'll be one on each of the centromeres, and they'll be facing out, and it's a protein structure that will attract the kinetochore of the microtubules that are coming from the centrioles, and that's how they will connect. And you can kind of see right there, that one's coming off and connecting, and that one up there. And as it connects, it will pull it in the direction of the centrosome, and as it pulls it in that direction, it will be met from the opposite side on the opposite kinetochore by another kinetochore microtubule that will stop it, and they will eventually even it out with tension on both sides somewhere in the middle. So at this stage right here, we again have one pair of chromosomes, or two chromosomes, one pair of sister chromatids, or sorry, two pair of sister chromatids, or four sister chromatids, and four centromeres. Then from there, we go ahead and we move to metaphase, which is the longest stage. It takes about 20 minutes, give or take. And in metaphase, you can see that they have been, the chromosomes have been pulled to the middle with equal tension there, and you can now see the spindle, which is formed when those centromeres get to, or sorry, when the centrosomes are on the opposite ends, on the poles, away from each other. And then that midline, that's kind of an imaginary line where they pull them to, is called the metaphase plate. And it's kind of represented by that dash line, the red dash line right there. And we have our total here also. Right here, this also just shows you that the centrosome is at one spindle end. This one, and then the other one's at the other spindle end. Our total here, again, is one pair of chromosomes, or two chromosomes, two pairs of sister chromatids, or four sister chromatids, and four centromeres. And from that step, we then will move down to anaphase. And during anaphase, that's when where the arms of the sister chromatids were held together by the cohesin, it is actually separated at that point so that they can be pulled apart and towards the pole end of their centromere, or sorry, yeah, their centromere and the centrosome that is pulling them. And they go centromere first in that direction. At this point, when they've been separated like this, they are now daughter chromosomes. They are individual chromosomes. So the what were chromatids, two sister chromatids hooked together forming one chromosome, are now individual chromosomes of their own, and we no longer will have what you would call sister chromatids inside the cell. So our new total is 4 chromosomes and 4 centromeres. And they will head over to the last two steps. We kind of put them together because once the one happens, the other one follows pretty quickly, the telophase and cytokinesis. Here you're going to have them start to form their own individual cells. And you'll have the cleavage. And you can see the cleavage furrow right there as they start to pull apart to form 2 individual cells. You'll also see formation of nuclear envelope and nucleolus is forming. You can see the centriole and centrosome that will go with this one, and the one that will go with this cell down here. So with telophase, we will have 2 daughter nuclei form. And with the cytokinesis, we will end with 2 daughter cells, 2 separate cells. So that's kind of the gist of it. This is just the real basic, trying to follow it through with just this specific number of chromosomes so that we can keep track of the number of chromatids. Obviously there is a different number in the actual cell division. But I hope this helps, and comments or any help would be great so that we can go ahead and make it better. Try and work on the drawing next time.